Clinical research is the study of human volunteers or participants so as to add to medical knowledge. It is not routine medical care. Clinical studies can be divided into two types. The first type is observational studies, where participants are assessed for biomedical or health outcomes. However, the researchers do not assign any diagnostic, therapeutic, or other intervention or treatment. Examples are cohort, case control, and cross-sectional studies. The second type is clinical trials or interventional studies, where researchers do prospectively assign participants to receive a diagnostic, therapeutic, or other intervention or treatment, to test whether it works or not. The participants could be assigned to one or more treatment groups, or arms. The treatments could be different, or the same but at different doses. There may also be a control that does not consist of the treatment. It could be an inactive, harmless substance or placebo, a procedure or surgery that omits the therapeutic step, or no intervention at all. The definition of an intervention or treatment is any process or action that is the focus of a clinical study. The action could be receiving a drug or drug combination, a biological substance such as cells or genes, a device, a procedure or surgery, a therapy such as chemotherapy or radiotherapy, a vaccine, or other product. The intervention could be an investigational one or one that is already available. The thing being applied could be non-invasive, such as education, diet, exercise, or preventive care. In a clinical trial, the researcher assigns the participants to the intervention in either a randomized or non-randomized way. The groups could be parallel, so that, for example, half the participants receive A and half receive a placebo at the same time. In a crossover trial, the same participants who are given the first treatment switch to the second treatment, so they act as their own control. And in a factorial trial, combinations of two or more interventions are tested at the same time. So participants are assigned to receive A and B, A without B, B without A, or no A and no B. When testing a potential treatment, you first perform preclinical laboratory tests in vitro, such as with cell cultures, and then in vivo, in animals. Before human testing, you need to submit evidence and proposed protocols for approval by the National Regulatory Agency. Then, there are a maximum of 5 clinical trial phases during which you apply for approval to launch and sell your product. The stages of the trial are phase 0, 1, 2, 3, and 4. These phases are based on definitions by the United States Food and Drug Administration, FDA, but are accepted around the world. The trial phases differ mainly by study objective, number of participants, and duration. The first phase is early phase 1, also known as phase 0 or a first-in-human trial, and is not required by the FDA. This is a small-scale study that tests the drug's target in the body, what the drug does to the body, and what the body does to the drug. Only about 10 to 15 volunteers are given a very small dose of the drug, which is subtherapeutic and not expected to treat disease. The drug is given for only a week because this phase is a pilot or exploratory study that does not yet look at drug safety or effectiveness. A phase 1 trial tests drug safety and toxicity in fewer than 100 volunteers, in groups that receive gradually increasing doses to find the maximally tolerated dose. Phase 1 trials last a few weeks. They do not have a comparison group, so they are uncontrolled trials that determine the drug's most frequent and serious adverse events, how the drug is metabolized in the body, and what represents a safe dose. A phase 2 trial tests a drug's short-term effectiveness in a few hundred patients receiving either drug or placebo for a few months. This proof of principle study can be a controlled or uncontrolled trial. It tests whether the drug treats the disease and allows you to obtain a dose-response curve and to study positive and negative side effects. Phase 3 trials are large-scale studies of drug efficacy and safety. They are conducted in hundreds or thousands of patients in different study centers who take either the drug or a placebo or other treatment. These trials normally last a few years and are often randomized controlled trials that determine the risk-benefit ratio and continue to record side effects. After regulatory approval is granted, Phase 4 trials test longer-term cost-effectiveness and safety in thousands of patients in different places and possibly different countries. The objective is typically to study the economic benefit of the drug versus the standard treatment over a few years and beyond. These are post-marketing trials that study long-term risks and benefits and optimal drug administration. Medical devices follow a similar path, starting with a small-scale pilot or feasibility study of safety in 10 to 30 patients, followed by a pivotal trial in a few hundred patients, and then long-term post-approval studies.